So welcome everyone to the third day of the Genome Design and Analytics short course run by the UCSD arm of the Center for Biosustainability at the Novo Nordisk Foundation. The topic for today is going to be the Bitome, which is a data structure that we found to be very useful for genomic data analysis. And initially we were using this largely for looking at trends within genomic information, but we've really started to accelerate our capabilities for machine learning using this genomic data to predict a variety of different phenotypes, everything from expression to gene regulation, transcription start sites, and I'll be stepping through a number of those examples. So in part one, I'll just be talking about what information is in the genome and how do we quantify that and perform kind of statistical analyses, look at trends, and that type of thing. So once again, I wanted to give credit where credit is due. And so we have a really nice sequence analysis team here in the lab. This effort was kind of kicked off by a PhD student in the lab, Cam Lamro, who, along with Dr. Paulson, conceived of and implemented this idea for the Bitome, as well as another postdoc who was part of that original paper as well. And he's really done a lot to extend this from the statistical analysis to the machine learning. And a lot of this work will be, especially the code, will be his work that we'll be talking about today. Also on this team was a master's student, Sujit Chu, who did a BSMS in the lab and built a machine learning model of TRN structure, which is going to be one of the case studies that we're going to be stepping through. And then Patrick as well has been heavily involved in doing mutation analyses, and which is a lot of inspiration for why we want to do DNA analysis in general. And we have a really good team of undergraduates who have been rotating in and out of this lab over the last, I guess it's almost 2 years now, as we've been developing these tools. Okay, so just an outline of this first part of the talk. We're really just going to be talking about the basics of DNA sequence, especially our focus is going to be on prokaryotes. I'm going to briefly mention eukaryotes, but almost all of our applications, or all of our current applications, are in eukaryotes. We do mostly E. coli, but we've extended beyond E. coli at this point, although I'm not going to go into any of those cases in the case studies I go into today. Everything will be E. coli based. But it is interesting and important to contrast with the information that's in eukaryotic tissues or eukaryotic genomes, just to kind of foreshadow where this is going and some of the challenges in making these kinds of data structures as generalizable as possible. Then we're going to be talking about different data types that you can layer on, whether they're experimental or kind of conceptual. So organizing genes by regulons and stimulons and the TRN, which are concepts that we introduced at the last short course, which really had a heavy focus on doing transcription regulatory network analysis of RNA-seq data. And then kind of culminating in what do you actually get for grouping all of this information and organizing it? So what happens when you can mathematically represent all this genomic information and start to look at trends? And that's this data structure that we call the Bitome. And I'll be showing some of the example analyses that we've done so far, kind of displaying the statistics of information in the genome and how that kind of provides a more basic understanding of what is actually, how your organism is storing information in its DNA. And then this will lead directly into machine learning, where we're taking that quantification of information and trying to predict the function of the DNA based on that. So, so that's just an outline of this short course section. So as I'm sure we're all aware now, we are sequencing data is the most rapidly growing data type in biology because it's so cheap and reliable compared to other large-scale omics data types. And so the number of new genomes keeps increasing. We keep sequencing different strains of E. coli. We evolve bacteria under stress conditions and then resequence them. So there's a ton of DNA sequence data that is becoming available. And so the challenge isn't any longer measuring what the sequence is. It's understanding what it means when you have one sequence versus another. And a lot of the focus of today's topic is not necessarily on the coding regions, which is a whole other animal entirely, but really thinking about the non-coding regions and how does regulation work and how does transcription initiation work and what kind of functions can non-coding DNA have and how do we quantify those functions? Yeah, so the challenge now is to understand the information within all these large-scale data sets as opposed to obtaining them or processing them in those types of bioinformatic kind of tasks. So what is in the DNA? So just focusing really on the region around a gene, very broadly, you have some kind of promoter region upstream of your transcription start site that controls the expression of a gene. You have where transcription starts. You have the mRNA that will result from transcription. At some point, you have termination of that RNA. And then, this RNA has some kind of information in it. And so this whole unit is called the transcription unit. And it's really a nice organizing principle beyond just where are the genes, because as we'll see, there are transcription units that can contain multiple genes in bacteria. There are alternative transcription start sites that can have different contexts that is still being determined. So, so there's not this very clean kind of textbook, one gene, one promoter, one mRNA kind of understanding. Instead, there's quite a complexity here. So organizing these transcription units is one really useful principle for making sense of mRNA expression data and these different data types that we'll be talking about shortly. So in prokaryotes, the promoter region looks something like this, at least for E. coli, and we're looking now at the specifically for a sigma factor called sigma 70, which is how the polymerase gets recruited to begin transcription. So sigma 70 helps to recruit the RNA polymerase by binding these two motifs, the -10 and -35 box. You may be familiar with these kinds of consensus sequences from a molecular biology class, but for E. coli, it favors this TATAAT -10 box and the TTGACA -35 box with roughly a 17 base pair spacer between them. And it's important to note that these are general trends. This is not a hard rule that the sequence is exactly this, and exactly 17 base pairs apart. In fact, it's hardly ever the case that these are exactly what the sequences are. There's almost always some distance from this, but this is, if you took an average over all the promoters, these are the consensus sequences that you would find. These are the average, most frequent sequences that you would find. And so, so sigma 70 likes these sequences and helps recruit the polymerase to the site, and that helps to initiate transcription. And different sigma factors can have different binding specificities, and that helps to determine where in the genome they get recruited to and which genes they actually help to initiate transcription for. In eukaryotes, you might be familiar with this Tata box as part of the core promoter region, but there's also additional upstream regions that help. There can be enhancers quite far away from the promoter. In general, it's a much more complicated picture. This is, I'm mainly just showing this as a compare and contrast. We don't really go into any kind of eukaryotic analysis here, although I would say that the literature, people are really starting to do a lot more with trying to build machine learning models based on eukaryotic promoters to predict activity, and they seem to be making a lot of ground. So as you might be aware, the genes in bacteria are often organized into operons. So operons are going to have multiple genes that are all within a single transcription unit. So you can have one promoter that then transcribes multiple different genes that have some kind of intercistronic spacer. You might have different Shine-Dalgarno sequences that are recruiting your ribosome to begin translation, so a ribosome binding site. But in general, this is yet another complicating factor, is that these transcription units can involve multiple genes, and promoters and terminators can vary between different alternative transcription units. So it's important to keep this in the back of your mind, that this more complicated operon structure is present with alternative transcription start sites, alternative transcription units. And these are measured if we look at mRNA, if we do transcription start site profiling, transcription termination profiling, we can identify the starts and ends of all these transcription units and detail these in an experimental way. I'll talk a little bit later about building machine learning models to try to understand why certain sites are transcription start sites or termination sites, etc. And in eukaryotes, the additional feature that you have is these introns. You still have a 5' untranslated region, 3' untranslated region on your mRNAs, but your mRNAs also have exons, which are coding, and introns, which get excised out to form your final processed messenger RNA. So even though all of this initially gets transcribed, it's then spliced, all your introns are spliced out to make your final coding transcript. And there's a lot of regulatory reasons for why alternative transcripts exist in eukaryotes, but in fact, bacteria, fortunately, we don't have to worry about that because it's yet again more complications. So just an example of how this works, you have these different exons, and alternative splicing can mix and match them to make different versions of a protein. So in this case, you're just removing these introns to form your final mature RNA. And yeah, different combinations of these exons will produce different protein products. Just to give more context for what kind of genomic content is in these transcription regions. But really, we want to focus on this is a very helpful diagram because we're going to keep going back to these concepts. And this is the recruitment of the bacterial RNA polymerase to the promoter region. And you can see there's quite a few different subunits involved in the RNA polymerase complex. The critical one that I mentioned earlier is the sigma factor. So RNA polymerase can bind to different sigma factors. If you're binding to sigma 70, that sigma factor is going to be recognizing a -10 and a -35 box. Sometimes a -35 box isn't even present, and oftentimes when a -35 box isn't present, Instead, you get an extended -10 box that can interact, and just this region alone can recruit the polymerase. There's a core recognition element that has some sequence identity that helps recruit it. There's a discriminatory region that is thought to actually play a role with a regulatory molecule that can directly bind to RNA polymerase called PPGPP. So, and then there's just this last piece here. There's an upstream promoter element that can be enriched in A's and T's that can interact with this alpha subunit in kind of a non-specific manner. So, so there's quite a lot of what I guess what this is mainly showing is that there's a molecular biology reason that the identity of these sequences matter. It's because they're directly interacting with different proteins in a shape recognizing fashion. And so if we can quantify the sequence information in a way that represents how well these are binding, we can start to predict whether or not a promoter is going to recruit the polymerase well, or if it's going to recruit it poorly, just based on the DNA sequence. And so in the first half of this lecture on the Bitome more broadly, we're going to be talking about, how do we collect all this information and quantify it. And then in the second part, we're going to be talking about how do we actually learn what makes a good sequence or a bad sequence for the function of the DNA, in this case, the function of the promoter being to bind the RNA polymerase. Yeah. And so just to build on this point, this is really, you can really think of the promoter as the key regulatory region, and it's all about what it says to the RNA polymerase. And so I already mentioned the role of the sigma factor, which helps the polymerase recognize the binding site and initiate transcription. You're probably also aware of transcription factors that can also bind. Basically, canonically, they would bind upstream and help to recruit the polymerase potentially. But oftentimes, they bind directly on top of the sigma factor recognition elements, the -10, -35 box. And in these cases, they might be repressing it because they're sterically hindering the RNA polymerase from binding. Although sometimes they can, for example, bind in the spacer region and help bend the DNA and help to recruit the polymerase, help the DNA be in a shape that the RNA polymerase likes to bind to. So there's really quite a lot of complexity in the interaction of different proteins that bind to the promoter region that either help or hinder the RNA polymerase from getting recruited to that site. And the result of that is, the regulatory function of that promoter. Is it constitutively expressed? Does it require a transcription factor to be there? Does it require a transcription factor to not be there in order to initiate transcription? It really comes down to a combination of all these factors. But the exciting thing from an analysis perspective is again, going back to the sequence, we can understand where these are binding and why, because we can identify motifs that these proteins like to bind to, that represent steric interactions that are favorable for those proteins to the DNA. So if we add together, this was the topic of the last short course, but if we add together all of these kinds of regulatory interactions, the transcription factors binding to the DNA, we can get these regulons, which is a transcription factor that binds to multiple different promoters. This was the topic of the ICA lecture, was actually determining what these regulons are in a top-down manner, working backwards from RNA-seq data and understanding which sets of genes move together, and then inferring which transcription factor is co-regulating all of these genes. You can think of today's lecture as moving from a more bottom-up perspective. We know that there's a transcription factor that's binding somewhere, And instead of working backwards from the expression data, we work forwards from the DNA sequence and say, can we understand why these transcription factors are measured as binding to certain locations? And can we understand what their regulatory activity is? So can we interpret the function of that binding based on the DNA sequence? And so by doing so, we can actually get a DNA sequence basis for the entire transcription regulatory network. And so one of the case studies that I'll be going into in the second half is a machine learning model that we built for predicting the TRN from the DNA sequence alone. So just reading off the DNA sequence, predicting which genes are regulated by which transcription factors, which is kind of the ultimate culmination of this understanding. If you understand what the motifs are, where they are, which ones are, and how activity relates to sequence changes, you can actually quantitatively predict the regulatory activity of different promoters. So this is a diagram illustrating a concept that we call the metastructure. This is an older name for what we're calling the bitome in this paper. But what the metastructure really is a collection of genomic data sets all layered onto the same genome sequence. So we know that we can sequence the genome of our lab strains. Maybe we have some sequence variants, but we can also get a whole bunch of other genomic data. For example, RNA expression from RNA-seq. We can figure out where we can measure peptides. We can get transcription start site profiling. We can look at Operon and transcription unit structure based on transcription start, transcription termination sites. And as we layer all of this information on, they start to become interoperable. They start to say things about each other. So for example, you might think that you have a promoter, an active promoter, because it has a really nice -10, -35 box. So you can look at your mRNA data and see, is it actually expressed? Or do you see a transcription start site there? Or if you see a transcription start site, can you read how long that transcription unit is by looking at the mRNA? And oftentimes you'll see different transcription units of different length stacked on top of each other, and you can see these discontinuities that kind of give you an idea of where the transcription unit is starting and stopping. So it's helpful. The bottom line of this is that our early metastructure work made it clear that it's helpful to not just look at these data sets separately. It's helpful to layer them and look at them in context of each other. And that's when a lot of these statistics really emerge. And that's when you can do a lot of the quality filtering and get these bigger picture stories about how transcription units and transcription regulation work. Just some more context for what kind of experimental data can come into this. So a lot of these data sets just look like these little peaks where you're measuring transcription start sites, RNA-seq. You might find ribosome binding sites from ribosome profiling or from a computational analysis. We can now do terminator-seq. And all of this can be mapped onto and integrated in terms of a DNA sequence. And so you can see some of the statistics here for this particular data collection, how many different of every data type were layered onto this genome. And it's clear that these are pretty large data sets, right? You're dealing with hundreds of thousands of each unit, and so it really behooves us to do something sophisticated with our analysis. One, we want to look at trends very broadly to try to glean some large-scale understanding of what these data sets are actually telling us, because it's just too much to just go into on a case-by-case basis. Even though in a lot of our early efforts, there was quite a bit of manual curation of these genomic data sets to figure out what data points are believable and what might be problematic. And two, to turn this into a predictive model, clearly you need some kind of rigorous machine learning, because it's just too much to figure out on a case-by-case basis. But I'll talk later about how do we actually get interpretable models and still make sure that we're not ending up with a black box when we have thousands of underlying models and we don't really know what's going on because the data sets are so large. And so the metastructure kind of became this concept of the bitome, and the difference between the two is that the bitome is really a matrix. The bitome is all of these data types that I was mentioning, everything from, at the simplest level, the base, a binary yes or no is a base pair within a gene. So is it part of a coding sequence? What is the cog, which is a classification of the ontology of the gene? So what is it doing? Which codon is it a part of? We can quantify all types of things, and then we can put them in a giant matrix, which is quite sparse. And you can see here, it's quite sparse. That just has all this information, and it can be largely binary. A lot of this data is binary here, and so that the concept of this binary information matrix became the bitome. So you have bits, ones and zeros here in this matrix. And once we have this matrix, we can start to do statistical analyses, and we can start to do machine learning. And that's really where we've taken this. And you can see here in panel E, this is from this paper in NIR that we published on the bitome. The amount of information is quite different across bases. The main differentiator is kind of obvious. It's whether a gene is whether a base pair is within a gene or not. But there are some surprise regions where there's quite a lot of data that maps to that and functions and things. And you know, it's interesting to see how much information, how much overlapping information and overlapping functional responsibilities certain base pairs have, right? And there's all kinds of basic constraints on information kind of considerations that can be extracted from this. And we've really just begun to scratch the surface. I would say we're not even close to really understanding what the limits are on packing information in the genome. So just more data types that are in the bitome, or more discussion of what these are. In addition to the raw sequence itself, we can map on any observed mutations. So these can be base pair substitution frequencies across all strains of E. coli. So you could say how frequently are certain base pairs mutated, or it can be adaptive laboratory evolution mutations. So these are mutations that occurred when the cells were subjected to a particular stress. And those mutations then should be conferring some fitness and therefore have some functional importance. And that can be a way to, by determining which base pairs within a genome mutate in order to increase fitness, that can give you an idea of which base pairs are functionally important, right? Because they can induce an improved phenotype on certain conditions. And so it's yet another interesting data type to cross-reference with other things. We can also map onto the genome the protein structures, and these protein structures have their own annotation, binding sites, active sites, dimer-dimer interface regions. All kinds of things have been annotated in databases like UniProt, and these can also be imported and layered onto the bitome. 2D structures as well, hairpin structures and mRNA we found them to be quite interesting and important, often showing up in these adaptive laboratory evolution experiments as regulatory. So destabilizing or stabilizing a hairpin can affect how much the translation efficiency of those genes, and have a regulatory role there. As well as transcription regulatory network membership. We can look at pathway membership in metabolic networks, all kinds of things. So all of this information is kind of knowable information. So there's no error associated with this. It's just functional annotation. And so these are the bits, the ones and zeros in the bitome. This would be contrasted with something like a binding strength or a motif similarity, which is a continuous metric. That wasn't, those types of functional annotations weren't in this first round of the bitome, but we've been layering them on afterwards ever since for some of the machine learning applications, and we found that to be quite useful as well. So this is what the Bitome matrix looks like. It's not this small, it's much larger than this, but just a quick snapshot of it. It is a binary matrix, and you just have locations. So this is genomic location. It's spatially organized within the genome from base pair 1 to base pair 4.6 million for E. coli K12, which is the length of the genome. And then you just have different genomic content here. So at the simplest level, we have what is called a one-hot encoding of the sequence, meaning out of the 4 base pairs, only one of them has a non-zero value. So this first base is a G, the second one's a T, so on and so forth. And you can see we're in a genomic, we're in a gene region here currently. So gene is 1. It's a coding sequence as well. You can see this is part of this TGT codon. So there's a G and a T there, so on and so forth. So you can, this is essentially what it looks like as you zoom in. It's just a binary matrix of ones and zeros. It can be quite large, but with sparse matrix handling, it's not too bad to deal with. And this is just another snapshot of what information is in the in this version, this initial version of the E. coli bitome. I don't think it's the final version, but it is the data sets that were readily available and how many of them there are. So there's 10 to the 5 mutations that are currently annotated. This is just the number of genes in E. coli, so that makes sense. Several thousand. And then a variety of different other genomic data sets are like where the ribosomal RNA is, how many ribosomal binding sites have been identified, how many terminators have been identified, how many transcriptional attenuators have been identified, so on and so forth. So quite, so on a log scale, you could see some of these are, there's hundreds, some there's thousands, but it's all encapsulated within that binary matrix. And so what can we do with that? Like now we have this matrix, what can we do? Well, one of the quickest and easiest things we can do is just look for correlations among our columns and see, are there features that are correlated and have some kind of relationship to each other. For example, you could look and see, do mutations tend to occur at attenuators? In particular, you can separate this by different mutation types, so SNPs, deletions, substitutions, mobile elements, insertion sequences, whatever it is, and look for some kind of correlation. You can do the same with any kind of RNA secondary structure, and basically look for any value that is statistically significant here. One of the advantages of organizing this matrix spatially is that you can actually look for spatial trends in the genome very easily, and maybe that's inspirational for what might be going on. For example, it's known that that the termination region of the so there's in the genome organization, you have an origin of replication where replication starts, and then you have where replication terminates. And these are called the orienter regions of the genome. And there are some trends that are known, like there's very few genes in the ter region. They tend to not be very expressed. And regions on near ori tend to be your a lot of your high expressed genes, like your ribosome, are around there. And so you could look for those things. And here you're looking at different mutation locations across the genome. mutation frequency, and mutation type density, and color coded here. And so maybe you see some trends that might be interesting to you if you think that maybe there's some link or biological reason why certain regions would mutate more than others. For example, through connections to replication or other functions of those regions. So just some more quantitative analyses of what we can pull out with this type of data structure. Here we're looking at a motif matching, and specifically those motifs that I was talking about, the sigma 70 motifs for -10 and -35 box, what we call the Pribnow box and the -35 box. So the Pribnow box was this TAAT. You can see here, this is a positional scoring matrix, what we call a logo plot of the motif. And so the way you read this is that there's the bigger a letter is, the more bias there is for that position to have that base pair. So you can see at the first base pair, it's almost always a T and hardly ever anything else. Whereas at the fourth and fifth base pair, it slightly prefers having an A there, but there's really not a large bias towards these, and there's some alternatives like T and C there that can be used instead. And then you can see the -35 box is a relatively weaker motif where only the initial two T's are very strong, and these other base pairs have quite a lot of diversity. And so we can use these as a scoring matrix and say how far in front of a particular promoter is its -10 box from the canonical motif. So we can take a log odds score, for example, and just score if you have a T there, it's multiplied by the likelihood of a T, and you add it by base pair, and you get a score for how quote unquote good that -10 box is, or how good the -35 box is. And you could say, okay, I found the best -10 box in front of this transcription start site, so I think this is where the -10 box is. But it's not necessarily at -10. It can vary, and so that's what we're looking at here, is where are the best motifs for each promoter in E. coli. And so in this case, we're looking at the forward start and the reverse start of the Pribnow box, and you can see that they're more or less, there's a 2 base pair shift here because it's not taking this, it's the start and not the center of the motif. But you can see it's around -10, like there's a peak at -10, but there's quite a lot of diversity. Anywhere from -6 to -20 can be the best -10, quote unquote -10 box. It's not really a -10 box in that case, but there's a peak around -10. And -35 is the same, but even noisier, which is probably a result of the motif itself being more diverse. And so already, just from this big picture overview of where the -10 box and -35 box are, we can already kind of revise our textbook understanding of how a promoter works. It's not always just here, like here's the sequence of -10, here's the sequence of -35, and nice and clean like that. There's quite a lot of diversity in where these boxes are. Now, why would that matter? Like, why would it matter? Well, we don't know yet, but maybe boxes that are perfectly at -10 and perfectly at -35, maybe those are the most active genes. Maybe those are the ones that recruit the polymerase best. Or maybe if the spacer is a little bit shorter, there's a twist to DNA, and maybe the DNA isn't oriented properly for the sigma factor to bind, and so the DNA polymerase or the RNA polymerase can't bind there, and it's not as active as you would expect that gene to be based on purely sequence information. So so this is just, you could think of this as just a quantification of the genomic information, and we're not going to figure this out by hand looking at these on a case-by-case basis. We really have to plug this information into a machine learning algorithm and figure out, does this matter or not for whatever phenotype we're interested in, whether it's expression, transcription start sites, regulation, whatever. This is just to show how diverse the sequences are and how it actually is much messier than the textbook understanding is. Just another trend that we were able to extract that I thought was really interesting. There's this canonical understanding in the literature that so when you're replicating your DNA, you have your leading strand and you have your lagging strand. And there's maybe not everyone is aware, but your DNA polymerase, your protein that that replicates your DNA, is much faster at what it does than the RNA polymerase. And so you can imagine that you always have RNA polymerases bound all over your circular chromosome, right? They're just doing their thing, expressing genes as your cell is living its life. But then once replication starts, you all of a sudden have this much faster DNA polymerase that is going along and basically knocking off all of the other proteins that are on the DNA in a very physical way, right? And there's the intuition or some prior evidence that a head-on collision, meaning if the gene was going in the lagging direction, you would have a head-on collision between the RNA polymerase and the DNA polymerase. Whereas if they're both on the leading strand, if the gene's on the leading strand, you're going to have kind of a coincidental collision. But there's an understanding that a head-on collision is worse. It can lead to damage, or it can lead to these improperly transcribed mRNAs, whatever it is. But anyways, there's this thought that that bacteria like to put their genes, their high expressed genes, in the leading direction to prevent this kind of conflict between the DNA polymerase and the RNA polymerase. And so we're interested, and we split up the genes by genes that are on the leading strand and genes that are on the lagging strand and genes in the terminus region, where leading and lagging aren't really clearly defined. And we looked at the gene expression of those genes. And interestingly, we do see that genes on the leading strand are substantially more expressed than genes on the lagging strand, which is consistent with that kind of understanding. Of course, it's still quite messy when you actually look at the data itself. There's quite a lot of variance within that. But if just looking at the medians alone, lagging strand genes, as expected, are lower expressed, which is consistent with that expected biological phenomenon. There's also these chromosome interaction domains, which are physical domains that the DNA can form. So these are like DNA structural domains, and we actually found that there was an expectation that things near the boundary might have different expression than things that are farther from the boundary. And it did seem like that was the case, but it is kind of a weaker trend. But it is still interesting to look at these kinds of large-scale trends in the data. Another interesting analysis that we pulled out of this kind of motif analysis, again looking at gene expression, was looking at the -10 box in more detail. And this was based on this idea that I was just describing, that some -10 boxes might be better at recruiting the polymerase than others. They might be better at inducing transcription than others because the sigma 70 likes them. It can bind really tightly, and that helps initiate the transcription. And so we looked at the most frequent in E. coli, the most frequent of all of the top variants of the -10 box, and then looked at their expression. And the nice thing, so one you could see that if you believe this these trends, and it's, if you don't think it's just noise, there's quite a lot of difference in the median. So it seems like there are really good and bad -10 boxes, at least for this number of promoters, which may or may not be statistically significant. It's fairly difficult to assess. But the other interesting thing is that our canonical promoter, this average promoter, just to go back here, this promoter is the canonical one. It's the, if you just average all the promoters, what is the most, like what do they average to? It's this one. That's actually the most active one, in E. coli at least. So there's a reason that, like, this intuition that canonical is good is actually borne out in the data. All of the TAATs on average are more active than these other types of -10 boxes. And so, yeah, this is yet again kind of a prelude to the kinds of machine learning models we can build based on gene expression, or based on DNA sequence, and in this case, predicting gene expression. So you can already maybe guess where I'm going with this, but if we were to define, or you could imagine we could try to predict gene expression based on the types of promoters, promoter motifs or motif strengths that these promoters have. And that's exactly where we're going. So after this initial Bitome effort, we've continued to layer on other data types onto this Bitome kind of understanding, Bitome kind of data structure. So I already mentioned the -10, -35 boxes, and we include spacer as well. But we've also started to map on quite a few different DNA shape features that really help to quantify additional, how to put it, aggregate information of the interaction of multiple base pairs together, because the DNA shape calculator uses a 5 base pair window to calculate a variety of different shape metrics. And so it's kind of it's a different way of summarizing the information in the genome beyond the raw sequence. We also included a bunch of genomic information, things like leading and lagging strand in these chromosome interaction domains, as well as different kinds of structure, as already mentioned, transcription unit structure. And so we have multiple different sigma factors in the Bitome now. So it's not just, even though sigma 70 is the constitutive sigma factor, it's the main sigma factor in E. coli. There's quite a few other sigma factors that could be used. And many of them have different recognition motifs, different spacer lengths. There are, RPOS does use, for example, the same motif as sigma 70. So not every sigma factor has a different motif, but a lot of them do. And so we're now accounting for these, and these can be quantified. So you can have basically a score for every promoter for every sigma factor in kind of a crosswise fashion. And then we can also construct motifs for all the transcription factors. And so I've already alluded to this, but you can get a motif score using a position-specific scoring matrix for a motif based on how often a base pair is found at a particular location. So the rarer a base pair, the less it contributes to your score, and the more common it is. So if you have the most common base pair, it'll contribute to your score the most. And we're extracting these currently from RegulonDB, which is this really nice resource for E. coli transcription that's been curated by people. This is obviously a major limitation trying to extend this to other organisms, but as a one example of a way to solve that is with the type of analysis we were talking about last time with ICA to identify regulons. You can actually extract motifs from those regulons and then work backwards from there. So as long as you can get a motif, you can calculate for a particular promoter whether there is a strong binding site for that motif, for that transcription factor at that promoter. Just to talk a little bit more about the shape that I was mentioning, there's something like 14 DNA shape features that we're now calculating. There's this R package called DNA shape R. And these are the shape features, shift, slide, rise, all kinds of structural things, minor groove width specific to and interbase pair features here and intra-base pair features here. So all of this is calculated on a 5 base pair window, and the average is annotated in a base pair specific fashion within this Bitome. And now all of this is continuous data, so it's not within that binary knowable Bitome previously. This is in this augmented Bitome that has all these additional features, because the goal of the augmented Bitome is really just build as predictive models as possible. So we want to put in as much information as possible, even if all of this has error associated with it, right? It's not absolutely true the way the Bitome is. So what does shape get us? Why do we care about shape? Well, it's an interesting concept to say, because intuitively it has different information than the sequence. The sequence somehow combines to make in windows to make the shape at a particular location. Well, we found this really interesting trend that suggests that shape has something to offer when it comes to interpreting how good a promoter is. And the plot here is shape versus location within a promoter. So this is an average of all E. coli promoters, all aligned based on the transcription start site. So -10 is the average of -10 boxes for all real promoters. And then also in red is random regions. So random, you can see, kind of looks like noise. It just gives you a baseline for whether or not there's a characteristic shape around a promoter. And the clearest trend at the top is just AT density. So AT density is a metric of the melting temperature. So GC-rich regions are harder to melt. They have greater binding strength, and unwinding is a big part of transcription. So you might think that that has some association. And you can see there's a really high level of AT density right at the -10 box, which makes sense because the -10 box is an AT motif, right? It's TATAAT. So of course you have a -10 box with a high AT density. So that makes sense. And it also makes sense that it's helping the DNA unwind there. But we also see things in the shape features beyond that. For example, if you look at the roll, there's this really sharp increase in roll right at the -10 box. And there's some, characteristic slide here. And if you look across, it shows up in quite a few of these features. And so what we're thinking is that in addition to just this kind of crude motif score, which just is an absolute distance and from the motif and doesn't really give any kind of interactions or structural understanding, this kind of shape feature could actually give us a finer resolution into what makes a good binding site for a particular protein. And so the protein we're talking about in this case is the sigma factor, sigma 70, but we've also done this for transcription factors, as I'll talk about in a second. Another thing we're interested in is correlations between these shapes, because one thing that we're curious about is just whether or not all of these shape features have the same information in them. But you can see while they're much more correlated than random, which is shown on the right, they're not perfectly correlated. So, AT density is correlated with roll, which is these two features we were noticing have a trend with -10 box. So maybe that's just due to larger correlation between these features. But there are quite a few uncorrelated features. And so these could be independent pieces of information on what that DNA sequence looks like right there that could be useful for machine learning and understanding what makes an active promoter versus a not active promoter. And we've done this for different sigma factors now, and so you can see sigma 70 trends, sigma 38 trends, sigma 32 trends. All of these can be extracted. And we've also done this for transcription factor binding sites. So now the protein we're talking about is not the sigma factor, and the motif isn't a -10 box. This is now a transcription factor binding to whatever it binds to. In this case, it's FNR, so it binds to this motif. And you can see near the edges, you might see some kind of characteristic shape there that might be indicative of what makes a good transcription factor binding site or not. And this was actually the original application of this DNA shape tool, was to analyze shape around transcription factor binding sites to try to help understand why some are strong and some are not. And we've gone further. So one issue with doing a shape-based analysis is you have 14 different features. What we'd really like to do is collapse them all into a single goodness metric. And so to do that, we use linear discriminant analysis, which is not that important to know. It's basically like a PCA, a principal component analysis that maximizes the agreement with a particular direction, in this case, whether or not it's a binding site or not. And we were able to identify shape scores that differentiate a good binding site from a not good binding site for quite a few different transcription factors. So OxyR, FOB, CISB, all of these are different transcription factors that we've identified shape scores for, aggregate shape scores that collapse all of this different shape information into just a single score that says this is a good shape and this is a bad shape. And so that's the type of analysis that we're doing here to incorporate all of this shape information. Okay, so that ends the first part of this short course. And that is just to summarize briefly, what we talked about here is all of the information that we've been packing into this matrix about what is in the genome. Right? Like, so for every base-banded genome, what functions does it have? What experimental data sets have been measured that annotate that site as doing something? What genome sequences have shown a mutation at that site? And then beyond that, what is the shape of these sites? And what is the functional annotation? Things like pathway use, cog category, all these kinds of things. And then I hope I showed you the types of things we can start to learn from this. And over, I was alluding to this eventual use of this matrix for machine learning for actual prediction, and that's going to be where we're going next. So for the exercise, I'm going to be going through the notebooks that Cam published in his original Bitome paper, showing how do you actually manipulate and explore this knowledge-based Bitome structure. So this is before we put shape in, this is just this binary ones and zeros knowledge structure. And so I'm going to go through this repository. Any questions before I go through that? All right, so let's get started. So this repository is public. It was published along with the paper itself. I think I'm still screen sharing. Okay, so hopefully you can see this. And the important thing to note is that there's a key limitation here. This is really because the data is spatially organized from base pair 1 to base pair 4.6 million. It's really specific to a particular strain. If you have any kind of reorganization across strains or anything like that, you can't really compare two different Bitome matrices of two different strains. And so so this first repository that we're talking about is a K-12 specific repository used for this paper. And it really shouldn't be used much beyond that. The analyses that we're talking about in the second half in this other repository, this string or S repository, uses an updated Bitome code that Cam also wrote called Bitome 2. And this, in the interest of making these analyses more multi-strain compatible, it does away with this organized base pair by base pair structure. So everything is no longer in order, and you don't construct a large matrix at all anymore. At least initially, you only do it right prior to building a machine learning model. That was just done to make multi-strain easier. So just to take a look at the original paper, if anyone hasn't seen this, this was published last year, called the Bitome Digitized Genomic Features Reveal Fundamental Genome Organization. And the goal of this paper was really to study the information theoretic properties of this Bitome matrix, to gather only the knowable, errorless data, the things that could be ones and zeros. You can see this paper here, or this figure here that I was showing earlier. To gather the ones and zeros, so that the knowledge that is has very little chance that it's wrong, right? Like functional annotations and things like that. And then try to infer if there are constraining principles on the information within a genome. And so in the course of this, there are quite a few of these different statistical metrics on the information within a genome. So distance from ORI based on the origin of replication and versus the average amount of information. All these kinds of things were quantified, and that was the major outcome. And so as part of this, Cam submitted this repository, which is a snapshot in time of all the analyses that are possible with this matrix. So I encourage everyone to check this out. The caveat is that you have to use an older version of BioPython because they got rid of the alphabet function, which is one of the things that this uses. So just be aware of that, that is the case. So when you're installing this, you just have to install it with an older version. Okay, so the main notebook that I wanted to go through was this summarizing the general content of the Bitome. So there are quite a few notebooks here. And so, yeah, this one's called Bitome Content. So if you want to follow along, this is the one here that you would try. Okay. And so in terms of packages, just matplotlib, numpy, pandas, scipy, and then Seaborn is another visualization package. So everyone should have these. And then this Bitome package is the repository itself. So hopefully nothing causing a problem for people. And it loads in this bitome.pickle. So it loads in this Bitome structure. And then you're just ready to go. So it's just, right now it's just saved. All of this data was collected over time and curated, different binding sites, everything, a lot of experimental data sets. And so the purpose of this code isn't really to construct your own Bitome. If you wanted to do that though, I would follow the methods of the paper. It's, the code really is for the analysis of the Bitome. So you can see here, just starting off, it just does a sum of the, so now you have the test bitome.matrix, which actually has that Bitome matrix, right? So that's the key structure to use. And we're just summing it to see how much information is how many, yeah, it's a histogram. So it's how many positions have a certain number of bits. So you can see there's very few positions that have more than, say, 20 bits. 25, I guess, is where it really falls off. On average, most have around 10 to 15 bits meaning pieces of functional annotation, any of these, any non-zero values. And then you can look at particular features. So you can extract features by name. So you could see there's base, there's transcription unit, origin of replication, there's secondary structure. So in this case, this is protein secondary structure. So things like alpha helices are annotated. For transcription factor binding sites are annotated. These are extracted from Regulon DB. Which amino acid, which is it? Which COG category? Is it an rRNA? Is it a Scheindel-Garno sequence? Is another annotated binding site as a part of a repeat region. So all of this can be extracted by name. And then there's a little function here to look at the coverage of different features. So now you can see we're reproducing the figures of the paper here. Yeah. So this is just a nice notebook to show how to use this data structure. And you can see, sequence coverage, nucleotide, 100% makes sense. Every base pair is a nucleotide. Interestingly, 91% of the genome is in a transcription unit, where in bacteria, where the genomic information is extremely dense. In a eukaryote, you'd expect that would be a much lower percentage. Although, if you believe the ENCODE project, they say that there might be like 70% or something that gets translated into mRNA somehow, but it's not clear what's background. Transcriptional chatter versus real, so on and so forth. But anyways, to continue on, kind of a massive percentage of the genome is in alpha helices, showing how dominant this structural fold is within proteins. Carbon metabolism, 9.1%, all kinds of things. So functional annotation, structural annotation, Scheindel-Garno sequences, actually the rarest, which is interesting. So it's not a very large motif, and you only need one per mRNA. So maybe it makes sense that it's such a small fraction. Okay. Then moving on, we're still, again, working with this matrix. And so now we're defining a couple functions here, one to calculate the information content of a particular matrix, and one to calculate a running mean. And with that, we can now do this type of thing. So we can look at the bits per base pair, the bit density, and we can track the distance from the origin of replication now in megabases. So the whole E. coli genome is 4.6 megabases. So you have 2.3 on each side, I suppose, here. And you can really see this. There's really only this one region that has more information with this running average. But it's, it's from 11 to 13. So it's maybe not a massive difference, but it still might be interesting to look at that region and see what's going on. And then some additional functions here that are from the supplement that are here you can see it's for different selected features, the ones I mentioned before up here, or these. So in addition to the total bit density versus distance, you could create these plots with any feature. So you can, for example, look at leucine density. There's a big drop in leucine right there. I don't know if that's an artifact. Looks like alpha helices. There must be no genes there. I wonder if that should be the terminus. Maybe it's the terminus. I wonder. Because there's no alpha helices there, so maybe there's no genes there. I'm not sure. I'm not sure what's going on there. But anyways, yeah, you can see, yeah, there's the origin. Oh yeah, so it's centered on the origin here. So so it's probably not the terminus. I don't know. I don't know why there's no genes there. You can see there's a bunch of fur binding regions here. all your rRNA is around the origin, as I was mentioning before. Your highly transcribed genes like to be around the origin. This is thought to be, oh yeah, actually, I didn't mention this point. This is another interesting genomic organization fact. But because you're under fast growth, your cell is almost always replicating its DNA. And so on average, you have more than one copy of your chromosome within a cell. And so the regions of the DNA that get replicated first are near the origin. And these are going to have, on average, the highest copy number of DNA there. So maybe it's like 1.5 on average or something. So that's like a 50% boost in the amount of DNA. And so the cell likes to take advantage of that and put highly expressed genes there. So things like ribosomal RNA, where there's a ton of within your cell, tend to be around your origin because it makes it easier to express a lot of it. That at least is the textbook understanding, and you can see it borne out here by where the RNAs actually are in the genome. So maybe that's one trend that's interesting to see. You can also look at bit density by feature type if you want. You could look at pseudogenes, for example, where they are, and you could see that the pseudogenes have, on average, much less information. Whether that's a matter of annotation or if they actually do less is, a matter of how the matrix is set up. Okay. And then we can do a variety of different amino acid secondary structure analyses as well. So we have, with the amino acids, we have whether or not they're nonpolar, charged, polar. We can do analyses there. With the secondary structure, these are the protein structures that are annotated. Again, these are coming from UniProt. So we have, beta bridge, alpha helix, these types of structural things all annotated. And you might look at correlations between these kinds of things. For example, there's known to be translational pausing in front of domains that are difficult to fold. And so if you had a ribosome binding data set, that could show where the transcriptional pauses are. You could cross-reference them with particular folds in your secondary structure and see whether or not that type of thing is actually occurring. And so we had a paper a couple of years back demonstrating that kind of principle. And yeah, you can generate a table from this showing the same kind of the features. All right, so that is the basics of exploring this kind of bitome structure, at least enough to generate the paper results. Sorry, I just restarted my kernel and everything. Then there were a couple case studies that I thought would be interesting to go into in the paper. Both of them are more machine learning than we've talked about so far, but I thought it would still be interesting to go into. So the first analysis, so for the machine learning, there's a couple additional packages that would be helpful to use. So for simple machine learning, as in everything up from linear regression, decision trees, random forests, support vector machines, we like to use this package scikit-learn. So if you haven't used scikit-learn before, it's a very extensive machine learning package. It not only has the methods, it has quite a bit of visualization. It has model selection, dimensionality reduction. It just has a very comprehensive set of tools for doing everything up to deep learning, everything up to neural networks, I would say. And then so we use this package quite extensively. And then for deep learning and neural networks, we primarily use Keras, which is part of the TensorFlow package. So you might be aware of this. But I don't have time to do a whole tutorial on TensorFlow, but we'll at least go through what Cam did for this paper. And we're still using these same packages, as well as some others in our latest machine learning work. So the goal of this analysis was to figure out whether or not there is any Bitome features that correlate with whether or not a gene is essential. So the basis for this question might be that you would expect that maybe essential genes are in a particular genomic location more often. Maybe they have certain codons more often than non-essential genes because they're highly expressed, and so they're going to use the most frequent codons to ensure that there's enough tRNA to translate them, things like that. There could be a number of different features that correlate with expression with essentiality. The question was whether the Bitome matrix could enable us to figure that out. And so that's what this notebook is doing, is setting up machine learning models to predict essentiality based on whether or not, or based on the Bitomic information. Okay, so here we're preparing a Bitome matrix to train a model. And you can see here it's not as large as the other, the full Bitome, but it's for the genes that we have, it's a certain number of features that were extracted to use in the training. And then there's a number of different things that are specific to the machine learning from here on out. So this is almost entirely a machine learning workflow. And so if you're familiar with that, then this will probably be very easy. If you're not, this will probably be very confusing. But unfortunately, I can't go into what all of these functions do in the interest of time. But the particular model that Cam used for this was a neural network. And so here we have some parameters, a training set, and a testing set. And then, yeah, then the target values. And we're using Keras to set up this neural network. The activation function is these rectified linear units. If you're familiar with neural networks, you can set the number of hidden layers and a dropout rate, which are two neural network specific metrics. So the hidden layers is the overall complexity of the network. Dropout rate is a regularization term. So the more you drop out, the more regularized the model will be. You can see that the loss function is this typical binary cross entropy. And then you have an early stopping function that also helps with regularizing the model. And the regularizing just keeps it from being overfit. So really nice function here. If you're interested in running a neural network, this is a nice implementation of a neural network. It has a good number of options that are very typical for an implementation. So nothing unusual with at least with these the basic parameters, but the fact that it has explicit dropout rates, early stopping, you can basically tune the model to make sure it's not overfitting, and by comparing your training and testing gap, you can tell whether or not you have a good model or not. Okay, so he runs the model here. So this is the stratified k-fold. So splitting into training and testing data sets. He runs a neural network model and then examines the performance. So looking at true negatives, false positives, false negatives, true positives, or whether or not it's essential. On the test set. And so with that, he gets the cross-validation that then is tells whether or not the model actually can predict whether or not a gene is essential based on the Bitome features. And then just some plotting. And this is just like a master function that runs those models. Okay, so this is the full, so I only focused on the neural network so far because it has more parameters, but you can see this 16 hidden layers, no dropout rate, and no L2 regularization, which is another type of regularization. He also compared it to quite a few other tools for vector machines, boosted trees, logistic regression. I don't have to go into all of them. And then now you can see the actual evaluation. So this is this master run models and plot performance function. Sorry, I'm getting lost. Here we go. Run models and plot performance function. And you can see the output here. So let's spend a little bit of time to understand what this is telling us, because this is how very typical for how we evaluate machine learning models for this type of work. So first off, we always run a variety of complexities of machine learning models in order to tell whether or not adding complexity improves model performance, or whether a simpler model could give very similar performance. So you always want to, you generally will run an increasing spectrum of complexity, and neural networks is almost always on the high end of that. Random forest slightly less, and then things like logistic regression are the simplest. And to assess performance, we look at two things. We look at the absolute performance. So in this case, the area under the receiver operating characteristic curve. We also look at how overfit it is. So we look at the gap between the performance on the training set and the performance on the testing set. And so if this is, I'm not actually sure if this is the performance, sorry about that, but if this is the area under the curve, you can see that there's quite a large gap from the neural network performance on the training set versus the testing set. That would suggest that it's rather overfit. What you would want to see is that the performance doesn't change very much between the training and testing set. And so if you're comparing across different metrics or different methods, you can look for the method that has the smallest training testing gap while still having a good training performance on the training set, right? So it's called, dealing with your bias variance trade-off is in the machine learning terminology. And here you can see the confusion matrices for the different methods. So you have the true class on the left and the predicted class on the bottom. And so if it's true, essential, predicted, non-essential, that's a false negative, right? So in this case, this add boost, you could see hardly ever predicts essentiality and kind of misses a lot of the essential genes as a result. It does very well at predicting non-essentiality, but it doesn't do that great at predicting essentiality. What you want is this diagonal to be as high as possible. Logistic regression, you can see, does substantially better. You can see the performance of the added boost distance is good. Where's the neural network? Yeah, the neural network also performs quite well there. Yeah, and so moving on then to oh yeah, sorry. He does some different optimizations of these models by changing the features, and also what's called hyper-optimization. So hyperparameter optimization is changing these parameters in a way that maximizes the performance of the model. So these are the parameters of the machine learning algorithm itself. So so this notebook has a good a good walkthrough on kind of end-to-end machine learning, and that's all good. So you can see optimization performance here, and the neural network performs well after all this optimization. That's great. Yeah, so now you can see after all the optimization, neural networks performing pretty well, less overfitting, right? So they're not dropping as much comparatively, and they have the best performance in isolation as well. So seems like they're performing quite well. The diagonal is significantly higher than the off diagonal, still having some problems calling some of the essential genes non-essential. Okay, so then the last thing I wanted to touch on is the feature importances, because at the end of the day, you want to interpret your model. You want to understand, so here's a lockbox, meaning a holdout set performance, and you can see that neural network does well as well as support vector machines. But what you really want to do with this type of machine learning is interpret it. And so at the end of the day, you want to look at your models and understand why they perform well and what they think is important for what you're trying to predict. In this case, we're trying to predict gene essentiality. And so what we're looking for is what features does the model like and think is important for being an essential gene. And so different machine learning methods have different levels of difficulty with doing variable importance. Support vector machines, you can actually just look at the weightings on the values. That's a very simple way. Neural networks, you can't just look at them. You can do things like looking at how often nodes drop out. You can shuffle your variables and see how much they hurt performance when you shuffle them. So if you remove a variable, it should hurt the performance if that variable matters. There's also a more sophisticated plot called a Shapley plot. It's like a game theory type approximation, seeing how important a particular feature is for an output. So there are ways to assess, even though a neural network is kind of a black box at a surface level, and you can't just look at it to understand it. There are a variety of ways to still do feature importance. But here, Cam is just looking at the coefficients of the SVM, because the SVM was a very good performing model here. And you can see, most of them are around 0, but if you filter greater than 0.8 here, so now everything above this now is grabbing the most important variables. So in other words, this good performing SVM model that predicts gene essentiality, he's grabbing what variables help that prediction. And this is the result. And I think it's pretty illustrative. It's a bunch of cog categories. I don't know what these cog categories are off the top of my head, but it certainly makes sense that certain cog categories, like translation, are going to be correlated with how essential a gene is, right? Metabolism, energy metabolism, whatever. There's going to be, or replication maybe is a good one. There's going to be certain functions that should be highly predictive of whether a gene is essential or not. And indeed, that's what comes out. This one is interesting, whether or not it's in the CRP, regulon CRP, whether or not it's in the CRP regulon, this regulon of the transcription factor CRP, is predictive of whether it's essential or not. That's interesting to me. That would suggest that this gene is this transcription factor is primarily regulating essential genes, or at least enriched in essential genes. And yeah, and here you could see just a nicer version of this. You could see essential gene values versus non-essential gene values for each of these cog categories. So the non-essential genes are essentially doing nothing for this, but the essential genes are much higher. Okay, so that's it for this notebook. I think this probably is good for the Bitome code. There's another, just a quick snapshot, there's another similar workflow here, looking at ALE SNPs, building another machine learning model for those. if you're interested in this, it's another expensive, really nice machine learning workflow that Cam put together. Maybe read about it in the paper and see if it's of interest to you, and then you can check out that notebook. So just to reiterate, all of these are in the public Bitome notebook. They all use this binary knowable Bitome spatially organized matrix structure. The Bitome content is kind of the basics of manipulating this matrix, and then I just wanted to highlight those machine learning workflows because I thought they were really nice initial applications of this type of thing. So yeah, I'll take any questions. Otherwise, I think we could probably break for 5 minutes and then come back to do part 2. All right, let's, I'll pause the recording and we can break for, okay, welcome back. Okay, so in the second part, we've already, in those examples, started to use machine learning to take advantage of the Bitome to predict different things. We're really going to formalize that in the second half of the short course session for today. I'm focusing on how do we build predictive models with genome sequence information, what kind of considerations come into play for building a successful model, and also what can we do with them. And I'll be covering these 3 cases: predicting gene expression, predicting transcription start sites, and predicting transcription regulation, which are the 3 that we focused on so far in the genome design group. So what I want to show here was that what we've really constructed here is the Bitome, which is all of the information mapped onto the genome, organized in a matrix or organized in a database. It doesn't really matter. But there's all kinds of analyses that can be layered onto this and built on top of it. So we talked about imodulons before. There's also the allelome, which is any variants that we observe across strains or across a species in a pangenome. So we're going to start to link the Bitome to these other data types. And one of the case studies I'll touch on later is actually using the Bitome to predict imodulons. And so I think in the future, we really want all of these layers to be interoperable, and machine learning is a great tool to make that happen. So this is a beautiful figure that Cam made to describe the concept of what we see as the vision for sequence-based machine learning, and we really see it as its own design, build, test, learn loop. And so in the upper left, we have the Bitome, which is all the information that we have. We might have some kind of sequence variants that we're aware of, whether they're a synthetic library of promoters or observed mutations across strains, whatever, but maybe we have some phenotypic data associated with those that we can try to make sense of. And then we also have RNA-seq data sets. So we talked about the precise data set last time, which is this highly curated RNA-seq data set. So this is the input. And so what we want to start doing is to build machine learning models that relate these quantitatively, to predict ones with the other, and specifically predict these other things with this guy. And so these are some of the applications I'll be talking about. We're using it to predict gene expression. We're using it to predict whether or not a gene is going to be in a particular regulon or have a binding site. And we're also doing other things kind of under the hood, like identifying new motifs and doing mutation analysis that I won't be talking about as much today. But the calling card of our brand of machine learning, or what we like to emphasize is explainability. Because at the end of the day, even if we have good performance, we really aren't satisfied with that. We don't want to have black box models that, you know, I have to say, just trust me, this works. What we want is to have models that we can look at them and say, okay, this makes sense. I can understand what the model likes and doesn't like about predicting this thing, and that it agrees with the prior literature. You know, maybe we could look at individual base importance, or maybe it's using genomic features, like I was saying, leading and lagging strand correlations. And maybe we're identifying some new features that we can then go and test and say, okay, no one knew this was important for expression, but the model really likes it, and we can go in and try to test that experimentally and see if it actually has an importance for transcription that no one realized before. And so, what's the ultimate goal? Is it explanation? Certainly that. It's certainly part of the point of building these machine learning models is to analyze data and say, yes, we understand why the expression is what it is. We understand how the promoter sequence relates to expression. There's a fundamental value in that, and we can do mutation analysis across strains and understand how ale is resulting in adaptive mutations that change expression levels. So there's certainly a data analysis component. But we also want to do design with it. We want to do prediction, de novo prediction, and cell design. And here we want to do things like, if we have a gene expression model, we should be able to in silico predict what a more active or a less active promoter will be, and do promoter design in silico for promoter tuning purposes, or change regulon structure. If we know what the sequence basis for a gene's membership is in a regulon, why is regulated by this transcription factor, we should be able to mutate that promoter and remove it from a regulon, or add a gene to a regulon without affecting other expression of that gene. So these types of design prospective prediction is a major goal of this type of work. Eventually, we're not quite there yet, but I think we're getting the tools to start to do that. So just as a backdrop, I thought it was interesting to think about how RNA. So sorry, I should preface this by saying that the first case study that I'm going to be going into is the RNA expression model. So what goes into control of RNA level? What we measure at the end of the day is the steady state mRNA level. But transcription is a dynamic process. You have some rates of transcription that has to do with the activity of this promoter, and you produce mRNA, and some of this gets diluted out by growth, not a very large amount. Some of it gets degraded. Data sets on RNA degradation kind of don't agree that well. So I don't know if we have a clean data set for that yet. But we can make relating it to our first lecture on steady state analysis, we can make a mass balance on our mRNA level, saying that it's whatever our transcription rate is, and then minus these factors that take away the mRNA. And so you could think of a predictive model that is trying to predict the transcription level to be some function of the sequence of the DNA here, so how well it can be recruited, and then the state of any kind of NAP is nucleotide associated proteins, so things like sigma factors, transcription factors, etc. So I just thought that this was an interesting backdrop, linking the mechanistic to the machine learning, and to make the point that we're trying to keep these things connected. We want our machine learning to be rooted in biology. And at the end of this lecture, I have some very preliminary but promising kind of mechanistic modeling of transcription that I see as like the ultimate, or eventually something that we're going to tie in more formally with machine learning. Okay, so this is the first application. This is Cam's research project. Once again, presenting Cam's work. But here we're interested in predicting gene expression from sequence features, especially the promoter sequence, but extending beyond the promoter sequence. So extending into the 5' UTR, potentially the terminated region, essentially whatever the machine learning model thinks is important for gene expression. And so the basic question is that even possible? Can we explain gene expression levels from sequence features? And of course, when I'm saying gene expression levels, I'm talking about across genes, right? So for a single gene across conditions, there will be changes in its gene expression level, but it's always going to have the same DNA sequence. So we're not going to have a prediction there. Fundamentally, what we're asking here is, if you look at an RNA-seq data set, there's orders of magnitude differences in average expression levels of genes. Some just are always very highly expressed, and some are always very low expressed. And so we're asking the question, can we understand why that's the case? Why some genes are highly expressed and others are low expressed based on their promoters? And can we ask a machine learning model to tell us that? And so if we were able to do this, this would help us do things like predicting a transcriptomic state for any new strain where we just get a genome. We could say, oh, this is a very highly active promoter. This gene's probably very highly expressed. We could understand how any kind of strain-specific sequence differences would impact expression, and maybe guess at things like why AL mutations matter. If we can say, are they affecting expression or not? And then we could even potentially use these kinds of models for design. So if we can understand expression, we should be able to change the sequence to affect expression. So very typical kind of workflow for doing this. We need to select whatever features we want to train the model on, and our target data, and then whichever conditions we want to use. We like to do a little bit of preliminary exploration, just seeing if we can identify any correlations between features and targets without any kind of sophisticated machine learning. And then we can train some kind of regression model to predict expression. And then, as I mentioned, we like to build white box models, not black box. So we always look to explain the performance in terms of the most important variables and see if they're biologically meaningful. And then repeat this after, if the performance isn't good, we kind of go back to the drawing board and see if we need to find new features, or if there's something wrong with the data set or what. Like we go into a debugging phase to understand how we can improve the model performance. So what are these features? So there's many different ways to encode a sequence. I described the one-hot method earlier, talking about the Bitome, where you just hard code the sequence with no kind of additional information, right? It's just base pair 1 is a G. And so you put a 1 for G in that column. And that's very nice to do that. It's very easy, but it has a lot of variables. One way you can kind of help out your machine learning is to collapse some of your variables into features that are aggregate features. So I already mentioned motifs as the classical example of this. So if you have a 6 base pair motif, you're taking the information from 6 base pairs as well as some location information and collapsing into a single variable. That simplifies the problem for your machine learning algorithm. It doesn't have to, I'd say that it doesn't have to rediscover the motif itself. It can just say, okay, I have a score for how good this motif is. Does this relate to your feature, your target value of interest? So we've, in addition to the one-hot representation of the sequence, we've also done a number of feature analysis, Sorry, do you have a question? Oh, no. In addition to the one-hot encoding of the sequence, we've also done a number of these what are called we call them classical features because it's textbook understanding of what DNA sequence features matter for expression. Things like -10 box motifs, transcription factor binding sites, 5' UTR length, and GC content is thought to have to do with the RNA secondary structure, which can be stabilizing and help expression, whether it's on a leading or lagging strand, where what the actual identity of the transcription start site base is, what is the length of the spacer, which has to do with the orientation of the DNA as it twists, the transcription unit length and GC content, and this chromosome interacting domain, which is this 3D DNA structural information. So we've over time collected these kinds of features and augmented the Bitome matrix with all of these. And there's really no reason to do any feature selection before this. We just kind of, anything that is talked about in the literature as potentially being important, we try to add it to the one-hot and say, does this help or not? And let the machine learning method algorithm decide whether or not that feature is useful. So that's our general approach. So this is beyond the one-hot. This is like a featureized model is what we call it. I don't know if that's actually a name. In terms of our data, we're looking at precise here. So this is precise 2.0. If you've been to iModulin DB, you can see what the iModulins for precise 2.0 is. This is the raw data for it. Specifically, though, we want to look only at wild type data sets. And that's because we want genetic identity between the strains. So if you have an ALE endpoint, you're going to have different sequences. And if you have a machine learning model and it's trying to learn what an expression value is, but there's different underlying sequences for each sample, that's going to make it much harder for it to learn that. That would have to be accounted for somehow. And no genomic knockouts, just to reduce the amount of perturbation. And because it's difficult for the models to understand any kind of expression changes due to these kinds of unnatural or genetic changes, right? It's trying to learn what the sequence basis is, but you keep changing the sequence. So I think we can account for this at some point down the line, but we'd have to account for it explicitly. So initially, we're just doing a wild type model, and we're extracting wild type data to do that. The next complication, I already mentioned that transcription unit architecture can be complicated in E. coli. There could be multiple transcription units for a particular gene. There can be different transcription units, some that include one gene, some that include both within an operon, things like that. And that was also very difficult to pull apart. And so for this first iteration, we're just looking at genes that are associated only with a single TSS, so that we don't have to worry about, for example, if you have multiple TSSs, you have multiple promoter reference points, right? So what is a -10 box? Is it with respect to one? Is it with respect to each? How do you say which, if you have an expression value of this gene, how do you associate with one of those transcription start sites in particular? It's certainly something we'll have to tackle eventually. There's no getting away from it, but in this initial run, it was more complexity than it was worth to try to account for. So we just filtered out these more complex cases and just looked at genes that had a single TSS. And this is just what the expression distribution looks like, which I think is helpful, so you can understand what you're trying to train to and whether it has enough diversity to change. And one of the interesting discoveries, I don't know if it's a discovery, but I mean, we did this analysis looking at the the variance in expression across genes versus the variance for a single gene across conditions. And what we found was that the variance across genes is much, much, much larger than the variance across conditions, meaning that the majority of variance in expression is inherent to the promoter, not something to do with the regulatory state of that gene, which is a very important finding for this model to work. If gene expression was dominated entirely by regulation, and it just has to do with what the transcription factor states are at that time, then this model would probably not work at all. But because the basal level of expression changes so much across genes, there actually is something inherent in the expression that we can try to learn. And that's the basis for this. Another interesting analysis that we did was looking at expression within a transcription unit. So one of these operons has multiple genes within a transcription unit. We compared the expression of this gene to that gene. And on average, there's much less variance across expression of genes within a transcription unit, which would mean that there's some similarity. You would expect that if every time this transcript was expressed, both of these guys were expressed, you would expect that they would have the same expression level. In reality, it's not even close to the case, but at least it's better than random. At least it's less variant than random. So again, this suggests that there's something in the expression data that we can try to learn. So this was one of the initial efforts. Cam's done various versions of this model. This was a random forest version that tends to perform fairly well, but tends to get overfit a little bit also. And here we're just looking at the promoter region from -50 to +10, which encompasses that core promoter -10, -35 box, upstream promoter region, core recognition element, and end up with an R² of 0.59, which is not too bad. Another interesting thing was, so we, like, as I mentioned, we always want to look at the variable importances to figure out what the model likes. And it was interesting to us that the most important feature was the identity of the transcription start site base pair. And this is actually, we looked in the literature, this is a known thing that this matters. So it was nice to see that. We also saw -10 box base pair show up. This is purely a one-hot model, by the way. I should have mentioned that. So this does not have those additional features. I'm going too far back. This doesn't have those additional features where we're feeding it a -10 box and saying this location is important, incorporate this in your model. We're just feeding it the one hot. And you can see that it pulls out that this base pair in the -10 box is important, and this base pair in the -35 box is important, as well as some in the upstream promoter region. So the initial one hot model was actually pretty promising without really helping it out at all. And just showing this type of thing, now mapped onto, so this is showing this type of thing now mapped onto this structural diagram again. So I'm kind of aligning the location of the binding site with the variable importance plot from a model. You can really see how the base pairs that are important have annotations. So like this -10 box base pair is right on top of that. You could see that there's this kind of surprising amount of importance at the TSS. So some -35 stuff. The other surprising finding to us was that there are some base pairs in the middle of the spacer that seem to be important, and I think that that's an underexplored thing. But by and large, the expected -10 box, that big T there, showed up, and so there's this concordance between the variable importances and our expected biological understanding. The other thing we try to do to tell whether our model is performing well, other than looking at the variable importances is to look at our training test gap. I already mentioned this before in the previous, when I was walking through CAM's other machine learning workflow. But we run multiple versions of a model, and we see training R squared versus testing R squared, and see how much they actually drop. So for example, if you just look at your training data set, you might see that this neural network is performing really well with the one-hot model. But then if you look at the test data set, it drops all the way from, like 0.95 to like 0.2, meaning that this model is horrendously overfit, and you probably don't want to use it without adjusting the hyperparameters. However, something like this support vector regression here has a much smaller training test difference. It doesn't perform as well as this, but it performs at 0.6 on training and 0.4 on testing, so not too bad. That's not bad of a train test gap, and so that would suggest this model is probably generalizable. It's not too overfit to use in new conditions. If you wanted to use a more complicated model, you would have to incorporate additional regularization. You wouldn't want to just use this model. Yeah, so this is kind of just saying the same thing. We always test a variety of complexities, and then you get a split here. So yeah, this is more or less saying the same thing as the last problem. You can do this testing not only methods, so elastic SVR, random forest neural network, but also you can change your data set. So you can look at different normalizations of your data. So you could take log2 data versus absolute data, or you could use different feature sets and just show if there's kind of a sweet spot for performing well or not, based on the training test gap. So the neural networks do some inherent training test gap within their fitting procedures. So not all of this is completely independent analysis, but it's still good to see this as a reference, and you can kind of select, maybe you select this point because it performs so well. This all standard log with SVR. It goes from 0.8 to 0.6, pretty good performance, not too much overfitting. So you could use this for model selection. Okay, so I wanted to get back to this point on raw sequence versus summarized features. So everything, the models I was showing there were a lot of one-hot models. So with one-hot, you just encode it like so. If it's a C, you just put a 1 there for C. Otherwise, whatever. And this is a really good thing to try when you're building a machine learning model, because you're not biasing your model by prior knowledge. So if there is something that is just pops out of the data, if you just give it a one-hot representation of your sequence and your model can find it, then great. Then it's probably real and you can discover it. These things can discover motifs, they can discover enrichments, they can discover base pair interactions. But these kinds of models are also very vulnerable to overfitting, because you've taken your sequence, which is already a lot of features. Your promoter region is like 100 base pairs, let's say. So for one promoter, you have 100 variables already, and now you've expanded into 400. So I mean, you're not making it easy for your model to not be overfit. So the argument for summarized features then is that they should be more generalizable. If you have overfitting problems, then helping your model out by saying, okay, these regions are important. Maybe you should look at the -10 box strain. Can address that issue. So they're complementary methods. And in my opinion, what we really want is a method that incorporates both and even uses machine learning to refine how we do motifs and things like that. Like get them to be more dynamically connected. But I think that's going to be a bigger challenge down the line, and not something we can immediately address. And then other end of the spectrum, going from, or maybe rather than addressing overfitting through these aggregated features, there's actually this unintuitive result that deep learning methods can actually end up, even though they have thousands of parameters, or I mean, some of these models have millions of parameters, but I mean, not the level we're building. And a great degree of complexity. They do internal simplification things that end up making them more generalizable than simpler models are. And so we've started to use deep neural networks. I'm showing convolutional neural networks as an example, but we're playing around with different types currently, because they have an inherent kind of collapsing function themselves. So if you're not familiar with the way a convolutional neural network works, it literally convolves your sequence into these features that then get learned and mapped onto a simpler representation. And so this can inherently collapse your sequence into fewer variables. And you can also feed it in as other channels, other features that you want to just feed it in. So even though we have problems with overfitting, even with simpler models, we're exploring deep learning because it performs unreasonably well at not overfitting, despite not having really enough data to train thousands of parameters, which is kind of unintuitive. But I just wanted to mention this because it's one of the directions that we're going in, and it relates to some of the examples that we'll be taking a look at in the exercise. So how do we build a convolutional neural network? In this case, the problem we're talking about is transcription site, star site classification, which isn't that important to know, but we just feed in the same promoter region that we've been talking about. And then there's a number of convolutional neural network specific parameters. So very similar to the hidden layers that we were talking about with neural networks, but now we have some filters that in a stride and just additional parameters, but very similar to the others. So the neural network kind of understanding where these more numbers is more complexity and slightly harder to train. But there's some sweet spot that you have to find through hyperparameter optimization to get this to fit. So that's another direction we're going in. But I really wanted to touch on this example here. Sorry, let me make sure I'm in the right place. Yeah, okay. So this is one way to deal with regularization, is actually to add complexity to the model, which is very unintuitive, but is one of the surprising findings that people are discovering with deep learning, is that you don't need the scale of data to train the models that would be expected given the number of parameters. The other way to deal with overfitting, or another way to deal with overfitting, is to build in more fundamental knowledge, build in more regularization. And the basis for this is that there's actually some things that are very hard for machine learning models to identify on their own. Very broadly speaking, machine learning is great at identifying smooth associations. So if you're trying to find a linear correlation, machine learning models will spit it out with very little data. But promoters don't have a clear linear relationship between sequence and expression, or sequence and whether or not it's a transcription start site. It's not like a nice linear function where a bunch of things add up and result in your output. At least not clearly. Although we've had a decent success with some of these things. Rather, there's if you look at the details of what's going on in any of these promoters, you'll find that they have types in nonlinear behavior. What do I mean by that? So by types, I mean there are distinct classes of regulation that can occur. There's like a type 1 and a type 2 CRP activation, and there's like a repressor 1 form, and these are distinct locations where these things bind, right? So it's not like a smooth gradient. It's a hard cutoff that it's either mode 1 or mode 2 or mode 3. And machine learning isn't inherently really well suited for doing this, especially not linear models that are going to try to create some kind of like a little bit of each of these things adding together, which really doesn't make sense biochemically, right? It's not like an addition of 3 different things going on at every promoter. It's like it's either happening or it's not. And there's also some nonlinear behavior in addition to this type kind of thing going on. There's nonlinear behavior, and what's the basis for this? So what I'm looking at here is this I think this is from a PNAS paper, but what we're looking at is gene expression versus the predicted RNAP binding energy. So how tight the polymerase is binding. And that's a factor of all these things we've been talking about, how good the motifs are, the -10 and -35 box motifs are. So a lower energy is going to be more binding. But what's interesting about this is that you have no, this is the negative of it. So this is high, better binding over here on the right. So at negligible binding energy, meaning terrible binding, you have no expression. That's great, right? But it doesn't increase linearly from here. It's just no expression for a while, right? So it's a nonlinear curve. Then you have a linear region. And then what happens here? Why does it start going down again? Well, the idea is that it's actually binding too tightly that it can't proceed with transcription here. So it's just locked in at the promoter and has a difficult time actually initiating transcription. Now, this kind of thing is extremely difficult for machine learning to learn. It has three regimes here: an insensitive regime, a linearly sensitive regime increasing, and then a linearly sensitive regime decreasing. So that's hard for a machine learning model to learn. So the question is, can we actually build in some of our understanding into the machine learning model to help it out, to help it get there and figure out, maybe it can fine tune the details, but if we can give it the overall structure, it might have an easier time understanding it. And so this is another, this is from, I think, the same paper or the same group on how you can actually account for these energies explicitly. So this is like a biophysical promoter model now. So now it's not machine learning yet until you try to parameterize these things. Maybe you parameterize them with machine learning, maybe you parameterize them with nonlinear regression or something. But you can actually explicitly represent these energies in terms of individual elements that we've already been talking about. So you have like a -10 box element, you have a -35 box element. And so you can start to imagine that maybe you build a machine learning model that just predicts these energies, and then you plug them into this and evaluate that performance instead, right? There might be some hybrid mechanistic machine learning model approach that we can use. And that's really like that's a regularization method that helps it figure out these kinds of things. Regularization meaning helping the model figure out more realistic structures. And so I don't want to go into this too much detail, but I just wanted to give an example of what is possible with this. So we took a look at the imodulons that for some reason for CRP has 2 imodulons, has a CRP1 and a CRP2 imodulon with very similar motifs. You can see there's not too much difference between the motifs. So the question is, why does it split into 2 imodulons? Well, I mentioned before in the example that there's types of regulation, and for CRP specifically, there's 2 classes of activation depending on where CRP binds. It either binds directly upstream in class 2 at -41, or it binds even further upstream at -61. So it has these 2 classes. This is known structurally, and the question is, do we have 2 imodulons for CRP because they represent these 2 modes of activation? So it's so that was the hypothesis, that maybe these are related. And so what we did is, again, using the Bitome information, we looked at where the binding sites were, and we saw that there were two peaks. The genes that were in the CRP2I modulon had a very clear peak right at, as expected, around -40. Whereas the genes in the CRP1I modulon had a binding site peaks that were much more around the expected -61, although there's some noise there. It's not clear if it's dimerizing or something like that. So by doing this kind of bitomic statistic analysis, we're actually able to get a mechanistic understanding of how the binding is occurring. And so that kind of foreshadows that the machine learning and mechanisms aren't that far apart. It's just a matter of figuring out a way to integrate them cleverly and mechanistically. And this is consistent with different mutations that have altered the CRP activity. I don't want to get too much into this, but the question is, so there's mutations that affect the activity of CRP, and some of them affect one of the CRPI modulons, and some of them affect both. And the question is why? And the hypothesis was that these two binding modes have different binding affinities. So one might bind at low activity and one might bind at high activity. And so we built a mechanistic model of this regulator, saying that one is a tight binding site, and one is a loose binding site, and that model was able to reproduce not only the curve here, sorry with the animations, not only this observed curve, the interaction between CRP1I modulon and CRP2I modulon, but also the behavior of the mutants, showing them that the mutants that only affect one of these will end up over there. So yeah, I just wanted to go through this example to foreshadow kind of the ultimate end goal of regularization is really going from black box machine learning to white box, and beyond that to mechanistic machine learning interface. With the time I have left, I want to give one more quick machine learning model that we're working on, which is the transcription regulatory network modeling. So here, the nature of the machine learning model was very simple. It was just a logistic regression model. So for classification, logistic regression is as simple as it gets. A low score is 0, and a high score is 1. And you just try to find a score that separates your classes accordingly. And what we're trying to predict here is whether or not a gene belongs in a particular regulon. So by using the promoter sequence, we can scan it for all motifs and say, is this gene going to be in a regulon or not? So for example, if it has an ALLR motif in front of it, then you'd say, okay, if it's a strong motif, yeah, sure, it's in that regulon. And you'd have a positive prediction for that. And then you can say, can the machine learning model predict whether or not for all of these regulons, whether or not a gene will be in the regulon based on its promoter sequence. And this is the result we had. We did it for 100 imodulons, all the regulatory imodulons in PRECISE-2. And we are looking at the area under the ROC curve. So anything at 0.5 is completely random. So this would be a terrible model. We kind of arbitrarily decided 0.8 would be our cutoff for what a good model is. And so you can see that ALR is a good model, ARC-A is not. We weren't able to find a machine learning model that could predict why a gene is in the ARC-A regulon. And so yeah, just highlighting some good and bad regulons. So what were we able to do with this? Well, quite a few things. One of the things we were able to do was to understand why we were getting so many FNR-related imodulons. So sometimes we get a single imodulon for a regulator, and it's like, okay, this is the imodulon that relates to activity. And sometimes we get multiple imodulons that all seem to be linked to a regulator in that they enrich for that regulator. And it's not clear why. Why do we have three different, well, we have two different FNR imodulons, but then we also have another mystery is that we have an FNR chip motif that doesn't even look like the FNR imodulon motifs. So it really wasn't clear what was going on with the FNR imodulons. And so we built models for 5 different imodulons that had some link to FNR. And we got 5 pretty good models for them. And so what we tried to do was look at the variable importances for what made a gene show up in this imodulon as opposed to any other, and see if that explains why these imodulons are showing up the way they are. And this is what's shown on the right. These are Shapley plots for variable importances, which is this game theoretic metric. So the way to read this is that higher values near the top of the plot are more important to the model. And if it's blue on the left, that means that a low value helps the prediction of being in the imodulon. And a red value on the right is the opposite, that a high value helps the prediction. So immediately what jumped out at us is this FNR plus IHF plus GCVB imodulon is very high in, well, one, you could see that IHF and FNR do show up, right? So that's good that their variables related to these regulators show up as important. So that would suggest this model is at least somewhat correct or meaningful. But surprisingly, LRP showed up as a very major influencer on this imodulon, which would suggest that this is potentially not just this regulator, but has to do with LRP. And we looked at a Venn diagram, and LRP has a lot of overlap with this imodulon as well. And so it could be a case where this should be classified as LRP related. For the others, it made more sense. This was called FNR plus NAR-L, and the 2 top features were the NAR-L motif score and the FNR motif score, which suggests that this is actually an interaction between these 2 imodulons showing up. And same with this FNR plus NAR-LP, we see a NAR-P score showing up here as very high importance, or the highest importance. So in other words, we were able to kind of figure out why there were so many going on. Is that this one is probably just misclassified and should be LRP related. This one is an interaction between two transcription factors, as is this. So and then these other 2, I don't know if we figured them out or if they're just solely FNR activity. But yeah, so this is one outcome of this work, is we're actually able to understand these complex regulon structures just by looking at feature importances. Another example of what we're able to do with this is to understand more complex promoter structures. So I mentioned before that RK didn't actually work that well. If you look, at least in our initial model, it performed really poorly, and meaning that the motif wasn't enough to explain why a gene was in the RK imodulon. So we looked at the motif by itself, and it's only this 5 base pair motif. It's kind of small, and it's not very specific. But when we looked into the literature, we found that it's the way RK actually works is that it has multiple repeats of this motif. It's not just the motif by itself once. It's there 2, 3, or 4 times. And so we recoded this into our matrix, into our feature matrix. And so we added 2 direct repeats, 3 direct repeats, and 4 direct repeat motifs, and saw whether that would actually fit the model. And you could see if we actually map this on promoters, you can actually see these direct repeats. So here's a transcription start site, and you can see a 4 direct repeat RK motif here right next to the chip binding site. So that's really nice to see. So you can actually visualize these direct repeats directly on top of the chip data, which is another benefit of the Bitome data structure. You can easily match these data architectures. So what happened when we added these direct repeats? Well, the performance improved. So now we have an ROC. We were down at 0.6 and now we're at 0.8. And if we look at the variable importances, you see the direct repeats are the model's favorite features. So it really likes these direct repeats, and that apparently is really what guarantees recruitment of RK as a regulator, is not just having the motif there, but having direct repeats of that motif. And so if you're thinking, how do I engineer regulons? Well, you would be sure to put direct repeats there if you wanted to make a gene regulated by RK. Okay, so that's the last example I had for today. I just wanted to summarize this really briefly. I hope I've convinced you that it's valuable to take all these different data types, experimental data types, as well as functional annotations like shape or cog category, anything we know about DNA, and map it onto individual base pairs to cross-reference them for analyses and for machine learning. Because once you figure out a way to quantify that, then you can do all kinds of predictions. And I guess I showed 5 examples, 2 from Cam's Bitome paper, and then 3 that we're working on in the genome design subgroup. And I hope I also gave you the impression that there's a lot of options for how do you actually quantify it, whether it's one hot or featureized. I didn't even get into, there's k-mers. You can count how many k-mers you have. You can auto encode sequences. I didn't get into all the possibilities because we're not exploring all of them currently, but they do exist. In order to go from base pair letters into a matrix with numbers in it to build machine learning models. And then you also have a lot of choices in how do you build a machine learning model. The answer of like what is the best method is always problem specific. We haven't found that sometimes neural networks perform poorly and sometimes they perform well, depending on how we depending on the question we're asking and how we set up the problem, and how we optimize our hyperparameters. And then another key takeaway is that in order to get a good performing model, a lot of times these models don't perform that well outside the box. And you have to take a look at your features. You have to take a look at your data set and try to understand how do you improve it? Like why is it not picking up that some feature that you thought was important should be there? Or looking at basic things like does your data have enough variance to train on? And is the variance due to sequence less than the variance due to conditions and things like that? So there's a lot of iteration and thinking that goes into building these models at this point. Of course, once they're better established, we'll probably have community standards and things, but we're still in a pretty early phase of building these types of models. And then last, this is my personal interest, but I'm always interested in making these models as white box as possible through variable importances and through linking to biological mechanisms everywhere we can, whether it's just looking at them and making sure that the variables that the model likes make sense, or whether we're incorporating mechanisms directly in our models through things like the direct repeats or things like the CRP case study, looking at different kinds of binding modes of the regulator. Okay, so thank you for listening, for being patient. That is the end of the lecture portion. I'll take any questions, but if not, I'll just proceed with the second exercise. Okay, so the second exercise comes from the Stringmore S repository, which is it's currently a private repository. I was a little wary about making it public because a lot of this is unpublished still. But if anyone is interested in using any of this, we're certainly making this available to any collaborators. It's just because all of this is public and getting posted on YouTube, I wasn't sure if we should make everything public yet. till we submit. But what is in this repository is two things. One, an updated Bitome package. So this is a total rewrite of the Bitome package that I just went into. The previous Bitome, just to reiterate, is this is all about this matrix. And the matrix is organized spatially from base pair 1 to base pair 4.6 million. The Bitome 2 is intended to be more flexible to enable multi-strain analysis, and because the large matrix can be cumbersome to work with if you don't need to use the whole thing. So Bitome 2 does a lot of the same things, but just doesn't assemble the matrix. And so it enables things like multi-strain analysis, and a lot of things are faster, and it just has, it's where we're doing future development right now. So if we take a look in the Bitome, you can see there's a large core, there's some machine learning, some utilities, and then some shape stuff related to DNA shape. Okay, we don't have to step through the whole package, but just to give you some ideas of what's here. And then the data, we have our genome, and then we have a bunch of genome features. So transcription start sites, transcription terminations. A lot of these are from Regulon DB. They're just tables that Regulon DB has, and the Bitome just uses. Okay, so on top of this, we have a number of workflows. So, and specifically, you'll see a lot of workflows here that are kind of deprecated. Some using the old Bitome 2, or so Bitome 1, but the key workflows are in Bitome 2. And you'll see we have the expression models here, we have imodulon models, and we have transcription start site models, TU finders, as well as some other things. And the other things, the most important one is the tutorial here. So I'm going to step through two things today. One is the tutorial, and the other is the TU finder that is using some of the more advanced features that have been developed lately. So now we're in the tutorial here. This tutorial is now using Bitome 2 code. So this is no longer the Bitome 1 world. It's a separate matrix, but or a separate set of functions, but a lot of the same capabilities are available. So if you're interested in starting off with this work, the main thing you want to get familiar with is the structure of the Bitome variable itself, because this is where all this genomic information is now captured. Instead of a large matrix, it's an object that has, attributes. Okay, so just to this exercise actually shouldn't take too long, but so we do our import. We import the Bitome, and this is a local import. This isn't anything that you have to pre-install. There's no pip install for this or anything. It's just local here. And then we import our genome sequence. So now we have this FASTA file of our genome for E. coli K12, and we can read off the information related to this file. So we can get the description that our genome is E. coli K12. MG1655, we can get the start of the sequence and the ID if we want to look it up on RefSeq. And then we get the GenBank file that has all the gene annotations, which is going to have more information for us to work with. So now this has, you can see what is in this gene table. It has our locus tags, gene start and end, whether it's forward or reverse strand, if it's a pseudogene, and its gene symbol. We can also get the same thing with proteins, but now we're getting the names of the proteins instead of the gene. Otherwise, it's more or less the same thing. And we can also get different features. So now you can see there's these different locus tags for this gene, and you could see, or sorry, for this genome, and it has a variety of non-gene genetic features annotated, things like repeat regions, mobile elements, non-coding RNA, Novel elements and repeat regions. Yeah. So so different other genetic features are annotated in the GenBank file that are not genes, and they're in this miscellaneous feature table. So that can be very helpful if you want to look for correlations between those and other features. Okay, and now we can actually build the Bitome. So so we create a Bitome object with this GenBank file, and we can start to populate it. So here we're doing it with the Proca GenBank file. You can also do it with the NCBI one. It's fine either way. So here we have the gene table head once again, and you can see now we have these kind of weird locus tags. Yeah, miscellaneous features. We have tRNAs annotated there. Okay, so now let's start to walk through the Bitome. That's all just important getting set up with our Bitome structure and seeing what's in it. So let's start to do some stuff with it. Okay, so we have the Bitome now building from the NCBI version. We're using a curated gene table and a curated protein table to help build this. And when we do that, we end up with this, this gene table, just like the one we already looked at. This is supposed to be broken. It's just to show gene table bad. It's just to show that it makes sure that your file is correctly formatted. So it's just a safe input. And you can also populate your Bitome here with other tables. And these are all those tables that we were just looking at in the repository here in the data here. So all of these get pulled in. Bitome 2 data example, there you go. And now you can see how much more information we have thanks to RegionDB. So now we have that there's sigma factor binding site here at this location. Or no, here's the location of it. Have the transcription start site. Have the Operon name. If there's no transcription start site, you get NAN. And you can take a look at all this information, but it's quite a lot. So you get quite a lot of genomic information just from these imports. And if you want to build your own Bitome, what I suggest you do is look at these tables and figure out how much of this data you actually have. If you don't have it, it's fine. The Bitome will work, but you know, it's obviously better to have more data. Okay, so now let's run some functions to explore this. So we created our Bitome table, Bitome many tables. Here's the Bitome GenBank that we're using now, just to find the sequence though. So you can use Bitome GenBank dot get feature sequence in order to get the sequence of this gene, and we're getting the sequence of B0001. You can also get a sequence just from the entire genome. So if you want the sequence from base pair 100 to 200 in the -1 strand, there you go. Okay, so this is just a nice package to work with genomic DNA in general. You can very easily grab sequences in different regions if you have those locations for whatever purpose. You can look at how far two things are apart from each other. This is a particularly useful one. If you let's say you have a mutation, right? You have a mutation at base pair 978, so pretty early in the genome. And you want to say, is there anything going on around that location? Well, you can do features in range and grab things below and above that, whatever region you're interested in. And you'll get a list of anything that's happening within that region. And so here it's just a bunch of genes because we're just grabbing this and not the fully annotated version with all the binding sites and everything. But if you did the version Bitome many tables that has all these binding sites, then this would be a much bigger list. But right now it's just finding all the genes in that region. We can also do visualization. So there's a Bitome GenBank view region. And here you can actually see any features mapped onto this. Again, this can get very messy the more features you have. So you might want to filter that first. But yeah, you can see what it looks like. Another really critical feature is, so I keep talking about motifs, right? So how do you work with motifs in the Bitome? So you have a PWM, positional weighting matrix, also called position-specific scoring matrix, and it reads in the motif. So there's a motif database that we have in our data file. Maybe we can take a look at that really quick. Where is it? It's in data. Okay, I don't know where this is. Give me a second. Here we go. Yeah, so in workflows CAD data. Yeah, motif PWM db.txt. So this is what it looks like. So we have the regulator here, and then we have kind of like a one-hot representation, but it's the log likelihood of being a particular base pair. So then this will be multiplied against your actual sequence to give a score for how much your base pair agrees with this index, and it does it for everything, and it adds them together. So that's the way it works. And so so here it's just importing the that database, and then it's grabbing the this particular regulator here, and then you're actually looking for. So yeah, it's just getting the position-specific scoring matrix, and then now it's using that position-specific scoring matrix to score all possible sites in a range. So this is n best matches 5. So this is the motif search. So it's giving the top 5 hits for the RHP regulator, the RHP motif, which is RHP here, this guy. So this is exactly what you want to do for motif searching. So you just change the range, create your PSSM for whatever your motif of interest is, and then you're ready to go. And if you were annotating this in the Bitome, maybe you put ones for these locations in your matrix. You can also look near a particular location. So if you're looking for example, the best -10 box next to a transcription start site, or the best RHP next to a transcription start site to find potential regulatory binding sites, you can just put in a single location. And that's enough. And then there's also this wrapper function, which is encompasses a lot of these functions, that is specifically trying to annotate all of these promoter features that I've been talking about. So this is a wrapper that does a lot of this under the hood, but under the hood, it's basically just doing this, doing motif searches on specifically the motifs for the -10, -35 box, but it also looks at the spacer length, and even gets the spacer sequence, and some upstream region AT density and downstream region AG density. So quite a few features that are of interest. The same features I was talking about for the gene expression model are constructed by this featureize promoter function. And you can make your own motif if you want, just by, you could feed it a list of sequences and then calculate a PWM. And why would you do this? Well, one of the things we found in CIGIA's transcription regulatory network model was that sometimes the Regulon that was annotated in Regulon DB, or the motif that was annotated in Regulon DB, wouldn't work very well for predicting whether a gene is regulated. It just it wasn't a very successful motif. But if we took the genes from the imodulon we could calculate a motif that was fairly robust and was predictive. And that so oftentimes we will take a set of promoters and try to build a motif for them, as long as we can identify the binding site. Now, in this case, you have to give it the sequences. If you're going to do this from scratch, it's probably better to use MEME, which is a can look for the motifs and score them. This just takes, like, these are all aligned perfectly, so it just does a calculation to transfer it from sequence to a PWM. It doesn't actually identify a new motif. Okay, so that's the Bitome 1 tutorial. Any questions on this before I move to the machine learning workflow that utilizes this? Okay, so there's actually two workflows that would be of interest. One is the gene expression model that I was talking about, which is, as you can see, a really extensive workflow, and we could be here forever talking about it. And then the other is the transcription unit finder, which is the more recent work that we've been applying these tools to. So let me first step through the transcription unit finder, just because I wanted to mention how nice we just updated the shape calculator. So I just wanted to mention how nice it is now, if anyone is particularly interested in that application. So much like in the other notebook, we import the same packages: matplotlib, numpy, pandas, scipy, seaborn, sklearn, and Bitome. So then we annotate the origin and terminus and CID boundaries. These are just not in the table yet. They'll eventually have their own table, but for now they're just here. And then we construct our Bitome. So we import all of these tables as well as these locations. Then we can start to do this initial profiling. So what I mentioned before is, prior to building a machine learning model, it often makes sense to just look at your variables first, see if there's any kind of correlation at all, figure out what you're actually trying to learn before just throwing it in the machine and letting it, try to figure it out for you. So in this case, what we're interested in is the intergenic spaces within transcription units. We're trying to predict, the overall point of this notebook is to try to predict where transcription units are, so predicting transcription unit start sites. And you could see here, the output of this script is just the intergene space within a TU. So in general, it looks fairly small, so there's not a lot of space between genes within transcription units. Now we can profile 5' UTRs, figure out on average what is the gap between the gene start, so where translation starts, and the transcription starts, where the mRNA starts. Okay, so so to do that, we grab the locations of the annotated transcription units from our Bitome matrix, and then we just sort it and calculate the TSS minus the first gene start. Okay, so it's just literally calculating that gap between where translation starts, which is the gene start, and where transcription starts, which is the TSS. I make a plot out of it, and you can see here that the average is, or the mode is like 40 or so, so not terribly long, but there are some very long 5' UTRs. So there is something potentially there with characterizing UTRs and trying to figure out what these longer UTRs are doing compared to the short ones. There's quite a lot of variance to try to understand. Not necessarily the most relevant for TSS finding, but it could suggest how likely it is, if you're scanning upstream of a gene start, how far do you have to go on average before you find the TSS? So it could be helpful that. So yeah, you can see Cam annotated that most of these are under 200. We can also look for transcription start sites within genes. and there's quite a few cases of that. So so within a gene, you would think that transcription would start upstream of genes, but there's oftentimes transcription starting within a gene, transcribing the next thing down. So there's no guarantee that transcription start sites don't occur within genes. There's 276 cases where they do occur. And yeah, to do this, he just identified where TSSs fall within genes. Yeah, here we're just using just finding motifs with -10, -35. There's some profiling here of where these actually are versus where the -35 and -10 are versus the TSS, but probably not that helpful to go into. Also here, there's sigma factor specific code. So for sigma and sigmas, so this is actually utilizing sigma factor specific motifs. Yeah, but let me skip ahead because there's maybe too much detail for this. So what I wanted to highlight was the shape code here, because now we have these 14 features within the network, and I was showing before those plots of the shape around the TSS region. And so we've been exploring whether or not we could use that shape to help predict the transcription start sites. And so here you can see the distribution of different shape features in these regions. And so you can see buckle has the center around 0, but some of these have kind of interesting characteristics that maybe could be learned by a machine learning model. And if you compare them to random ones, you'll see that a lot of these actually look very similar to random. So it's not clear if they're functional. But what we can do is plot this around the transcription start site region, and you can see again we're comparing the real TSS to random, and it substantially diverges from it. So you might think it matters, but some of these it doesn't. So it might not matter, right? And so it might look promising, but at the end of the day, we want to just add them as features to a machine learning method. Oh, this is too long to go into. But yeah, at the end of the day, we're plugging this into this other notebook here that actually utilizes all this data in a TensorFlow model to sorry. There we go. Yeah, here we go. So here we're running this TensorFlow model here, build CNN. So this is a convolutional neural network version of a transcription unit finding model. So it uses all of these sequences, and then and shape features everything. I think it's just using the one hot sequence right now, actually, but we want to put the additional shape and annotated features in there. And then it's again using this Keras package, but now as opposed to the neural network example from before, there's additional hyperparameters here, and filters, filter size, stride, valid, etc. So this is definitely too much detail to go into right now. So what I would say is email me if you want access to this. And I think we're in a point where everything is ready to share to collaborators at least. But yeah, some of it's still a work in progress and unpublished, so I don't want to just post it to the public repository. Just briefly, with maybe 5 minutes left, let me step through this expression notebook, which is the example that I was going into previously. So here you can see that we're now importing precise2, which is this precise expression data set here. Again, it's in the Strainware repository. Yeah, precise2 JSON here. So this is our RNA-seq compendium, and we can just use a nice function here called expression distribution to generate these plots of TPM, and then you can do log TPM, so log transformed, and generate these. These are all wrapper functions. So there's a lot going on under the hood here, but you know, at least if you have a new data set, you can easily apply it. And then we can set our one hot model. So there's this function called one hot encode TU promoters that uses a particular range. So you can say 50 pace upstream, 100 upstream, whatever you want, and it'll return a one hot encoded form of that sequence. And then you can do it to any region. So you could so one of the caveats is with one hot, it can be tricky because if you have any difference in your spacer, like I mentioned before, that -10 isn't always at -10. So it's hard for a one hot model to figure out that this -10 is actually at -6. Sometimes it can be hard. So so we actually do an alignment step to align the -10 boxes and -35 boxes across promoters, and then so it's one hot aligned, which makes it easier for it to actually understand that a -10 box is a -10 box. And then so here we're just setting up the same models: SVR, support vector regression, and random forest, and then you can get your plots here. So and then the last part is maybe the feature importances, which here's the one hot. So there's a function one hot feature importances that generates that graph that I was showing earlier. And then the other critical one is this feature importance function that generates the Shapley plot. So you can see that these more or less correspond to each other. Well, actually, these don't correspond that well. I think they're using different metrics. But the functions that Shapley cares about are going to be slightly different than just the raw weightings themselves, which is partly why Shapley is so nice. But you can see here that this -35 box base pair is important. The core recognition element right at the TSS is important, kind of as expected. So I think that those are probably the most critical functions to be aware of. So again, if you are interested in getting into this, just shoot me an email. The most critical notebooks are this tutorial, the expression, and then if you wanted to see the deep learning example, this TUFinder_CNN that has the convolutional neural network implementation. Yeah, but as you can see, with quite a few notebooks, the team is very busy putting these things together. Okay, so I think that just about wraps it up for today. I think we're probably good for summaries. Just maybe the last thing is just if you're interested in getting involved in this work, we would like to make the code more public. The Bitome code is a little bit outdated. A lot of these new machine learning workflows don't work that nicely. Some of the motif finding and things like that work better with the Bitome 2 stuff. So it really is the Strainware S repository that would be good to get familiar with. So just yeah, reach out to me. happy to share, happy to set up a follow-up meeting if you want to get involved with this. All right, thanks everyone for sticking through. Next week we'll be covering kinetics and thermodynamics. Zach Heyman will be helping, and we'll be covering his MassPy Python package for kinetic modeling, which he built from scratch and is really nice. All right, take care everyone.